It's a great pleasure for me to introduce our speaker today, Jake Vogel. I've known Jake for a very long time, since he was kind of at the stage that many of you guys are at. He took my class in his first semester as a rotation student at McGill when he was rotating through Sylvia Villeneuve's lab. So you might recognize Jake from his time at McGill. He's a good sort of homegrown success story. Well, it's not from Montreal, but I do recognize he's from Philly, I believe. Is that right?
Correct. Yeah.
Yep. So Jake's an assistant professor in the Department of Clinical Life Sciences in Malmö, the SciLife Lab at Lund University, and supported by the SciLife Lab and the Wallenberg National Program for Data-Driven Life Science. His training involved work as a research technician at the University of California, Berkeley, with Bill Jagist. He did a PhD with Dr. Alan Evans here at McGill. That's where you might recognize him from. And a postdoctoral fellowship, most recently, with Ted Satterthwaite at UPenn. He applies many computational techniques and statistical models to examine human imaging and multimodal data to better understand how neurodegenerative diseases progress and what may underlie them. The focus of Jake's work is involved in characterization of individual differences in disease expression. So many of his research pursuits are achieved through wrangling, I like that you wrote that, aggregation and fusion of large data sets with a focus on reproducibility and synthesis of complementary information. So thanks a lot for accepting to take the invite, and looking forward to talk, Jake.
Thanks so much, Millar. Also want to give a huge shout out to Millar, who was on my PhD committee and really was an MVP in many ways, beyond being a great scientist, also great mentor, great person. So you guys are so lucky you have him at the Douglas. And yeah, it's a real pleasure to sort of be back at McGill, albeit virtually, and giving the CSE talk. Definitely, as Millar mentioned, I've been in those seats in the Bowerman watching a talk. So it's really, really cool to be on the other side of it once again. And so, yeah, I'll talk a little bit about this idea of subtyping and staging of neurodegenerative diseases. Millar gave a fantastic intro, so thanks. And just to sort of reiterate some of those ideas, I sort of started this lab at Lund, the Dementia Multiomics and Neuroimaging Lab, or the DEMON Lab. And the focus of that lab is really disease progression modeling in the context of neurodegenerative diseases. And so what we're doing is we're trying to measure biological phenomena that are associated with the AD pathophysiological process and try to understand the sequence by which those events take place and model them. And by doing so, our overall goal is to try to forecast disease outcomes at the individual patient level, right? If we can model the disease, hopefully we can forecast what's going to happen in the future. We also want to resolve disease heterogeneity. This is really the major focus of this talk in particular. And we want to do as much as we can to try to learn something about disease mechanisms in humans. Now, this is, of course, very difficult because we're neuroimagers, we're working with observational data, but we do what we can. So I will get into sort of what I'll talk about today. This is interesting. I'm going to talk a lot about this idea of disease subtypes, the fact that in many neurodegenerative diseases, even though we tend to think about things as group averages, we tend to think about Alzheimer's disease is just a singular disease that we must treat. I'm going to try to convince you that there's actually a lot of different sort of subpopulations within a larger disease umbrella. And I'm going to be doing that in the context of Alzheimer's disease, but also in alpha-synucleinopathies and some other diseases as well. And I want to focus a lot in this talk actually on some of the clinical implications of these subtypes. I think the biological stuff is actually more interesting, to be honest, but the clinical stuff is what gets you grants. And that's kind of my focus now as a young investigator. So, but it turns out that stuff is pretty interesting too. So we'll talk about that. And I also want to talk about the idea of staging at the end of the talk, because even if you're not thinking about subtyping, it's important to understand where people are sort of along a different disease cascades and how we can sort of measure that and understand it. And then at the end, I'll just talk a little bit about, conclusions and some things that we're trying to do in the future. So I'm going to start by talking about Alzheimer's disease because the bulk of the talk will have to do with Alzheimer's disease. And I know that there's people working on all sorts of things at the Douglas, but just this will be a primer, maybe redundant for a lot of you, but hopefully a refresher if nothing else. So this is a very typical cartoon of the Alzheimer's disease pathological cascade. On the x-axis, you have sort of disease time from preclinical to prodromal to full-on dementia. And on the y-axis, you have sort of pathological changes. So basically measures becoming abnormal. And we have a couple of different lines here. The first one represents amyloid beta, which deposits into plaques very, very early on, actually in the preclinical stages before there are any symptoms. Then we have neurofib tau, which aggregates into neurofibrillary tangles. That comes along a little bit closer to what follows, which is the typical neurodegeneration that we see and brain atrophy that we see in Alzheimer's disease, which then is related to declining cognition. And so, of course, there's a lot more to it than that. There's lots of different processes that seem to occur along this pathophysiological cascade. Neuroinflammation is a big one, neurovascular dysregulation. All sorts of things are happening. But the big three are really plaques, tangles, and neurodegeneration. So the AD landscape has changed dramatically over the last really 2 years. And I'm sorry, I forgot to mention that really important for us as neuroimagers, we can actually measure these big three using biomarkers, right? So we can measure amyloid and tau using positron emission tomography or PET. We can measure neurodegeneration with MRI. And we now have a ton of plasma and CSF biomarkers that we can actually use to measure all of these phenomena. And really, everything I'm going to talk about now, with the exception of a couple of examples, will have to do with using these different imaging and sometimes fluid biomarkers to understand heterogeneity along this cascade. So the AD landscape has changed dramatically over the last 2 years, because now there have actually been some successful disease-modifying therapies that have reached significant results in slowing cognitive decline in clinical trials, and also doing so by, in theory, modifying some aspect of the disease rather than just treating the symptoms. And specifically, these disease-modifying therapies have been immunotherapies that have removed these amyloid plaques. The sort of rationale there is that the plaques build up very early in the disease, therefore that maybe they are causal to the other processes, and by removing them, you may slow down the other processes. So here's one of the results from one of the sort of 3 recent Alzheimer's disease clinical trials that use these anti-amyloid immunotherapies. And you can see that over time here, we're measuring amyloid here, so amyloid in the brain measured with PET. The placebo group does not change. It's very, very high over time. And the group that received lecanemab had a very significant and staggering drop in the amount of amyloid in the brain. So it works in that degree. It actually removes amyloid from the brain. And sure enough, there's several different outcome measures that have to do with cognition. And again, here you've got time, and on the y-axis, you've got global cognition as measured with this ADAS cognitive measure. And you can see, once again, the placebo group has a slower cognitive decline. I'm sorry, the group that was treated with this drug, lecanemab, that lowers the amyloid, had a slower cognitive decline compared to the placebo group. We're still getting cognitive decline here, but it is significantly slower, and that has been a huge success. Now, I want to highlight something here, which is the number of participants, right? These groups are they're pretty big, right? 850 people. At the end, they have about 700. And so I think that really emphasizes that these are very small effect sizes. If you need 800 or 700 people to get these kind of very small differences in the percent change, it really indicates you have a small effect size. And there's a lot of reasons why this might be, but probably the answer is that there's tremendous variability, right? They don't publish the actual data because it would look really messy. But the truth is that there's a lot of variability in treatment response. Why is that? Well, it's very interesting, because a lot of the previous trials that have tried to do the same thing have actually failed. And the ones that have been successful have very small effect sizes. And the most common explanation, which I think is a very legitimate one, is that we're intervening too late, that by the time that people get further along this process, some of the other sort of parts of the cascade are already underway and can't be stopped. And there's actually data to support that from these trials, where people that are earlier on in the disease process with less pathology tend to have better outcomes. It's also possible that we're just going after the wrong target, right? Maybe we should be targeting tau. It's more proximal to neurodegeneration and cognitive decline. There's also these other disease-associated changes. Maybe it's being driven by inflammation or vascular changes. Maybe that's the operative target that we need to look at. And maybe we just have no idea what's going on, which is still possible. But I think I want to highlight one other sort of main explanation that might account for what's going on, and that's the fact that there's such a variation in how people experience Alzheimer's disease, right? We're just not accounting enough for individual differences. We treat Alzheimer's disease like this one thing, and the reality, if you go into a clinic, is that every case is quite different, and the degree to which we understand those differences varies, but we at least know that they're there. And I think this is very important, because everyone's really all into this idea of individualized medicine now. And I'm really happy that that's the case, because I think that this is, we are sort of in a bit of a dark ages in medicine. And this is sort of like that's sort of the light at the end of the tunnel, where a person can come in and we really understand their genotype and their phenotype, and we know exactly what's wrong with them, and we know exactly how to treat them. And you've got this buzzword floating around everywhere, every single sort of, grant call, it's always there. And I think that this is a bit of sci-fi right now, because I think to be able to do that, we need to have just tremendous amount of data. We need to be having these huge biobanks that are starting to be collected around the world. And we need to have very sophisticated models to figure that sort of thing out, and a lot of good validation. But we don't have to fret, because we can sort of take stepping stones along that path to eventually reach this point where we're at individual level variability. And the first stepping stone along that path is subtypes, right? So the idea that this is the sort of first level of variation in a disease population. And this is really important, because this level of variation definitely exists. We can see it. We sort of know about it anecdotally, even without the statistics. And it's something that can be detected with thousands of data points, as opposed to hundreds of thousands or millions. And so this is sort of feasible under the current funding model. So I think this is a really, really great place to start, especially because there's plenty of examples in the medical literature of, just looking at subtypes and already getting a big advance in your ability to do sort of more personalized medicine. I think my favorite example is, and one of the simplest ones that we kind of forget about, is blood type, right? Blood transfusions are so important for so many different reasons. And, for a while, we couldn't really give these transfusions to everyone, because sometimes they would get rejected. And now we sort of just understand, okay, people have different blood types. If you match the blood type, then you'll have a more successful transfusion. And that had a huge impact on medicine. And really, there are only a couple of blood types in the population, right? It's sort of like a subtype. And so that's what we're looking at here. There's also been some examples in cancer, and more recently diabetes, and it's really promising. And I think that this is sort of the first step that we need to take in moving towards, this grand goal. So in neurodegenerative diseases, there seems to be two really big major sources of variation. And this is sort of visualized here by this cartoon that I'm taking from the Ferreira and et al. 2020 review in neurology. And I'm kind of adapting it here a little bit, but because I do think it represents the two main sources of variation in the population. The first is this vertical axis, which is representing disease severity. Some people get disease, you take a single disease, it doesn't have to be a neurodegenerative disease. I think this is true across many diseases. Some people get the disease much earlier, and they tend to have a much more rapid progression. Some people tend to get the disease much later. They have a much slower progression. And if you take an individual snapshot of a person, they could be sort of anywhere along this dimension, both if you think about across the population, but also within themselves and their future. So this is a bit complicated to disentangle cross-sectionally. And then there is variation in the expression of a disease, right? People could have the same underlying pathology, and it might express in different ways. It might affect different organs or different parts of an organ. It might come along with different symptoms, but the underlying neuropathology or just regular pathology is the same. So this is sort of what we're probably going to try to characterize if we're going to start doing subtypes of neurodegenerative diseases. And we're hoping that there are some sort of secrets locked there that might help us for clinical trials. And so this has implications for clinical trials, right? So what if we have some people who are fast decliners and some who are slow decliners, and we're accidentally enriching one of them for one of the treatment arms or the placebo arm with one of these groups, and we just don't know it? And that's what's driving our effect. And I think a much more important outcome is that what if some people just have a different underlying pathophysiology, and they're just not going to respond the same to the treatment? And if we can identify them, we can find subpopulations that respond really well, and we can treat those groups with confidence. And so the problem with this is that it's very hard to resolve cross-sectionally. We probably need to use data-driven approaches, right? We don't want to just hand pick and hand select things. The data can probably teach us a lot, but we have some problems. And I'll try to sort of give you an example of what I mean. So let's say, take an example where we have a bunch of neuroimaging data here, and it's various sort of cross-sectional data points, and we want to use some kind of clustering approach and figure out subtypes in this data. Well, the issue is that this data, the major source of variation in this data, right? If you think about it, they're just these three-dimensional, images with voxel values, and if you sort them, you're actually going to end up sorting them into these clusters based on just the amount of total pathology that's there, because that's the information that's in the image, right? People that have a lot of pathology are going to end up in one cluster. People that have very little pathology are going to end up in another. And so we wanted to cluster, but because of the nature of the progressive nature of neurodegenerative diseases, instead we end up staging. And that's a shame, because there actually is some variation here, right? There's a couple cases, for example, that have considerable occipital lobe pathology, and many of the other cases do not. And that sort of occipital spatial pathology seems to be in some ways orthogonal to the disease stage. And so it's that sort of orthogonal disease stage that is much harder to capture, and what we want to try to capture. So then along came Alexandra Young from UCL, who's just a fantastic researcher and a very, very close collaborator of mine. And she developed the subtype and stage inference model, or the SUSTAIN model. And that sort of combines linear progression modeling that's based on top of a sort of event-based modeling. And it combines it with clustering. So what happens in this case is, instead, this algorithm tries to assign a single sort of trajectory that fits the data, sort of linear trajectory from this cross-sectional data that basically creates a sequence of these images or of these biomarkers. And once it finds the best sort of sequence that fits the data, it then goes and tries to fit another sequence to try to fit more of the data. And this is really nice, because then you're actually, you're assuming everyone will progress along a trajectory, and you're just looking for different trajectories. And what's great about this is then you could take that model and assign people not only to a subtype, but to a stage along that subtype. So it's been really great to learn this algorithm, because it's a pretty elegant solution to the problem I showed you earlier. And so just by knowing SUSTAIN, I've been just kind of along for the ride, as many people have contacted me and brought me onto projects, or gave me data and asked me to help. And basically everything I'm going to present from here on out is kind of the results of those collaborations. So I think, because we're going to be talking about SUSTAIN this whole time, we should probably get into a little bit of the nitty-gritty of how it works. So let's imagine we have some biomarker, let's say it's MRI or tau PET, measuring neurodegeneration, and we have different brain regions. So the first thing is that we do need to assign these waypoints, okay? Because there is linear modeling that occurs between these waypoints, but we sort of have to, in some ways, inform the model of where it should be looking out for nonlinearities and so forth. So. And what the SUSTAIN model tries to do is it tries to come up with a sequence. So maybe, for example, region 1 will become slightly abnormal. Then region 2 will become slightly abnormal. Then region 1 will become even more abnormal. Then region 3 becomes abnormal, and so forth and so forth. And so there's this sort of inferred sequence of these events, and individuals are basically fitted to that sequence. And this is the best fitting sequence. Now, because SUSTAIN is sort of a Bayesian algorithm, you actually get a measure of uncertainty, which is really helpful. You can get an estimate of how certain SUSTAIN is in assigning someone to a subtype or a stage. Once this model is fitted to the whole group, you basically try to find a separate trajectory that can fit sort of some of the outlying data, and so forth, until you're not explaining the data very well anymore. So that's sort of how that works. And so the first place that we ended up using SUSTAIN together, Alex and I and a bunch of other collaborators, was here at McGill, when we looked at the progression of tau, which was measured using tau PET. So you can remember this sort of this cartoon I showed you earlier about the amyloid plaques, the tau tangles, and the MRI. Well, tau can be measured, as I mentioned, with tau PET. And this is actually something that came out very early on in my PhD, and my PhD was really focused on characterizing tau PET in Alzheimer's disease. And so during my PhD, we had, with some collaborations with people in Sweden, where I am now actually, we ended up aggregating about 1,700 tau PET scans across 5 data sets, of which we ended up using about 1,200 of them. And we applied SUSTAIN, the technique I just taught you about, to this data set, this large data set, to try to uncover if there are subtype trajectories of tau propagation in the Alzheimer's disease population. And so when we fit a single model to this data, we get something that looks a little bit like this. You have these sort of stages where the tau moves from the medial temporal lobe to the sort of temporal neocortex, eventually to sort of other parts of the isocortex, and finally, lastly, in sensory motor cortex. This is great because it looks exactly like the typical neuropathological staging of tau that we expect to see. So that was cool. But then, because SUSTAIN creates subtypes, we actually were able to find 3 additional trajectories of tau accumulation that looked ostensibly different. And we can sort of look at the average tau signal across each of these subtypes, and we get these sort of tau PET subtype patterns. And so here I'm showing the sort of average tau image for each of the subtypes. And in the middle here, I'm showing you the difference between one subtype and every other in terms of tau aggregation. So the first thing we saw was this sort of limbic subtype. and that really had most of the tau aggregation in the medial temporal lobes, and this sort of medial temporal lobe or hippocampal sparing subtype that was where we saw less tau in the medial temporal lobes and more in the isocortical areas. This was great because this is exactly what we expected to see. These subtypes have been described from neuropathological samples, and there's been a lot of work to try to characterize them over the last decade or so. But we also introduced these other sort of subtypes that appeared in the data, one that was sort of had a more posterior pattern that was involving a lot more of the occipital lobe, and another that was more lateralized, especially to the left side. And right, there's actually a visual variant of Alzheimer's disease, and there's a language variant, and they do actually have tau patterns that look like this. But these patterns were occurring much more commonly in the population than you would expect, given these rather rare clinical variants of Alzheimer's disease. So I'm not saying that there's definitely 4 subtypes. There's definitely only 4 subtypes. But from this and some of the sort of follow-up data we've seen with other data sets, these 4 patterns keep emerging. And so it is safe to say that there's probably some subtypes of Alzheimer's disease, at least. So they have some characteristics that are interesting. I won't go through all of these in detail, but just to give you some examples, this sort of limbic subtype had a much higher proportion of people carrying this ApoE E4 allele. This is a huge genetic risk factor of Alzheimer's disease. It's actually not uncommon. It's in about 20 to 30% of the population, and it increases your risk for Alzheimer's disease about 2 times if you have 1 copy, and about 8 to 10 times if you have 2 copies. And people that had this subtype were more likely to be carrying this E4 allele. They also had worse memory, and they were older. And then you have this sort of counter subtype, the medial temporal lobe sparing subtype, which sort of had some of the opposite patterns. So these people tended to be younger. They tended to have less likely to carry this ApoE E4 allele, and it was more of a dysexecutive phenotype. So it's important for us to sort of validate this beyond the single discovery data set that we had. And so we had a second data set called BioFinder 2, and we've been trying to validate and extend these subtypes in this data set. And it's really great because we had about 1,300 individuals, which is somewhat close to that huge multi-site data set that we started with. But here it's a single site. So we don't have to worry about any harmonization aspects or anything like that. And when we fit this sustain to this data by itself, really just fit the model de novo, or if we used the original model and then just fitted it directly to the new data, we got the same results, which is that we saw the same 4 subtypes, and they look really, really similar. What you're seeing here are spatial correlations between one subtype and all the others, and they're arranged such that the 2 subtypes or the 2 cohorts have the same ordering of the subtypes. You can see there's a really, really strong spatial correlation between the 4 patterns that pop out. We also found that several of the original findings were replicated, including the ApoE findings related to ApoE4, age, and the specificity of cognitive decline. And this is something that we're now just continuing to characterize. We've done a little bit so far. We have, for example, MRI data. So we're looking at longitudinal change in brain atrophy across these different subtypes. We also have several CSF biomarkers, and we're trying to understand if there are some other sort of molecular facets of these subtypes that can help us better characterize them and help us understand the underlying biology. But this work is actually really, really nascent right now. And actually, I'm probably going to put out an ad for a postdoc who will be working on this sort of thing in the next month or so. So if anyone out there is interested, definitely let me know, because there's some really cool work that we can do with this data. Another interesting way that this has been validated or is continuing to be validated is with these tau PET visual reads. So tau PET has now been FDA approved, and it means that you can sort of prescribe a tau PET scan. And when you do that, a physician actually, a radiologist actually needs to read the scan. There's nothing automated. There's no numbers they're looking at. They literally look at this image, and then they decide if you're tau PET positive or negative. And that is what is on the sort of label of this prescription. And you can't really, you basically have to do that. And so there's a group in Switzerland that was really interested in these subtypes and wanted to try to integrate the idea of tau PET subtypes into the clinical visual read. And so they've come up with this really interesting sort of decision tree to help a clinician evaluate if not only if an individual is tau positive, but then if they are tau positive, what subtype they are, right? And so you can see as you go along this decision tree and you make certain decisions, eventually you'll end up at one of these subtypes, S1, 3, 4, or 2. And it's been a really, really interesting exercise to work with these people in both trying to figure out what the sustained algorithm was doing and translate it to a human, but also stick within the requirements of the FDA approved a label for this drug or this prescription. And so that's been fun. You can see the sort of visual people have been, the same cohort has been assigned to a subtype by visual reads and by automated sustained subtyping, and they look really similar when you look at the grand average, but there are a lot of actually differences. Some people moved subtypes, and there's a paper that's sort of in preparation that's describing this and sort of describing why these differences happen. So this has been a really interesting move towards a clinical utility of this, but I think it's really important because, you know, it's going to really allow more people to be able to look at subtypes, and hopefully we'll be able to better understand them if we have more clinical data. Speaking of clinical data, I want to talk a little bit about clinical implications of subtypes once again. I already showed you this before, but I just want to remind you, you know, we're worried about things like accidental enrichment of fast decliners in a placebo or treatment arm of an Alzheimer's disease treatment. And we might also, of course, be worried about underlying disease pathophysiology. But I want to focus on this idea of people declining faster or slower in the clinic. It says that my internet connection is unstable. Are you guys able to hear me? Give me like a thumbs up or something.
Yeah, it's good.
Yeah, we can hear you.
Okay, great. Sorry about that. Zoom was messing with my head there. And yeah, so one of the most important findings we had from this original tau subtyping paper is that there were different rates of cognitive decline. So here you see sort of time from baseline at 0. And on the y-axis, you see MMSE, which is a measure of global clinical decline. And you can see each of these 4 lines represents one of the subtypes, and this sort of lateral temporal subtype seems to be declining a good deal faster than the other subtypes. And this posterior subtype is declining a lot slower. And so I've now seen 2 different data sets who have sort of tried to replicate this. One is one that we've done internally, and it replicated this lateral temporal subtype being the fastest. And another data set I've seen that actually was a full replication that found both the posterior being way slower and the lateral temporal being way faster. And that was a very large data set of Alzheimer's disease cases. And so we think that this is somewhat legitimate finding. And if so, it's kind of important when you're thinking about enrichment of clinical trials, possible enrichment with one of these subtypes or another one. And this is an idea that certainly I didn't come up with. Actually, Neil Oxtoby at UCL has been doing a lot in this domain of trying to define subtypes and see how they would affect a clinical trial. So what he did in this study, which is led by Cameron Shand, it's a preprint right now. They basically fit a sustain to a large sample of people that had MRI. And they found these MRI-based subtypes, typical cortical, subcortical, and no subtype. And then they took that model and then they applied the model to a new data set that was sort of like a clinical trial. And they found that in this new data set, using data that was not used to fit the model, they found that the same subtypes, that the 2 of the subtypes had sort of a faster clinical decline on either the CDR sum of boxes or the MPAC, both of which are sort of cognitive or measure cognitive or functional decline. And these effect sizes are definitely small, but it's interesting because it's the same subtypes they found in the discovery set. And this is just a little bit of a proof of concept that, you know, you can do the subtyping in your data set either post hoc or a priori, and have some indication of how that's going to affect the change in cognition over time. Another really cool example of this, also coming out of UCL from Danny Alexander's lab, is when they applied sustain to multiple sclerosis. So they had both diffusion-weighted imaging as well as T1-weighted structural MRI, and they used this data to create these subtypes, white matter subtypes, really cortical and white matter subtypes of MS. And just like we did, they found that the subtypes had a small but significant difference in their progression along the disease, right? So y-axis, again, is just some metric of disease progression. But I think what's far more important than that is when looking at treatment response in a clinical trial, they found that 2 of the subtypes responded very well, or at least more significantly, to the treatment, while another subtype really didn't respond at all on average to the treatment. And this is like the kind of the golden finding, right? This is why we're doing this, because we're assuming that there are some differences in the population, and those differences might actually be relevant to treatment of the disease. And so this is so we want to continue to try to get our hands on clinical trial data to understand if this is the case. but it's sort of a very promising example. Another example is something that in the context of not just a clinical trial, but also in real life existing memory clinics. So this is work that was led by Hannah Baumeister, who is in David Barron's lab in Germany. And so she had a really interesting cohort of like real prospective memory clinic patients, right? So 528 people that came into a memory clinic, either self-referred or referred by their physician at one of 10 sites across Germany. And of course, all of them were given a diagnosis, either MCI or dementia, or, if they could not find any objective evidence for cognitive decline, then they gave them a diagnosis of subjective cognitive decline. And then there was also 285 controls sourced from the same area. And so so all of these memory clinics, you go in and they give you, among other things, a neuropsychological battery and workup, and also an MRI. So this data exists in these secondary and tertiary memory clinics. And so Hannah applied Sustain to the MRI data from these memory clinics, and she came up with these two subtypes. One that's sort of limbic predominant atrophy subtype that really sounds to me a lot like Alzheimer's disease, which, by the way, I should mention, there was no Alzheimer's disease biomarkers for the majority of these cases. So we didn't know, it's a real memory clinic. We don't know if people have AD or not. So there was this sort of limbic predominant, and then there was this hippocampal atrophy sparing subtype, which had more of a cortical disease progression or neurodegenerative progression. And so, okay, fair enough. We got these subtypes. And just like some of the other studies, we found some differences in the rate of cognitive decline. Once again, here's the y-axis. We have cognitive decline, and these are these two subtypes. And the red indicates people who are later stages. The orange is people who are earlier stages. And so what we're seeing here is an interaction between subtype and stage. If a person comes in, we give them an MRI at baseline, and we subtype and stage them. And if we happen to find that they are of this sort of limbic predominant atrophy subtype, and they're a later stage, it seems like there's a much greater chance that they're going to have a marked decline on their global cognition. And remarkably, we actually found that same finding when we restricted the sample to only people that did not have cognitive impairment. Obviously, the effect size was weak, but the same interaction emerged. So that was pretty interesting. But if we really want, we have to kind of sometimes take things a little bit further beyond discovery and proof of concepts. We want to try to see if this would work somewhat in the wild, or at least replicate it in our data set. So we basically applied, we looked at the same thing in the Biophinder group, which is the group I work with here in Sweden. We are also a memory clinic. We also see patients prospectively by referral, and we also get an MRI. So we did a couple experiments where we fit the model in Delcode and applied it to Biofinder and looked at the outcomes, or we fit the model using Biofinder and applied it to Delcode. And we were absolutely blown away by the results. No matter which data set we fit the model in, we found extremely correlated staging and subtyping of the individuals. I mean, it was out of control. If we use the discovery set or the out of sample set to stage and subtype the people, we got almost the exact same subtypes. And once again, both a limbic and a hippocampal sparing one. And this was so good that we really didn't believe it. And we did a lot to try to break it. I tried, for example, running the entire analysis myself with my own code, not looking at Hannah's code. And we just kept getting the same result. So, I mean, what this means, when you see something like this, it's probably an easy problem to solve, right? There probably just is this variation. You get people who have Alzheimer's disease and people that or something like it, or have an amnestic or hippocampal kind of atrophy pattern, and people that don't. But it's very useful because those people, they do have different prognoses. And so, this is just something that was done in Hannah's German data set, but we're seeing, for the goal is represented on these survival plots, where we can have an indication if someone is at a certain stage and they're a certain subtype based on our MRI, we can give them an estimate with a certain amount of certainty about when they are going to convert to another stage of the disease. Now, of course, there is uncertainty there, but the fact that these models replicate so well, it gives me hope that we can actually find a lot of different memory clinics who can participate in this and probably end up with really, really similar MRI data and really similar subtypes. And we can start to narrow these confidence intervals, or at least make ourselves more confident in them. Because I think this is the overall goal. Someone comes into a clinic, they get an MRI, and we can give them an estimate of, the likelihood that they're going to convert to MCI or dementia over the next months to years. So this is sort of the goal, and we're getting a little bit closer. Okay, we're done talking about the clinic for now. Maybe you guys are really bored by that, but we're going to talk about subtyping in other contexts, and we'll move away from Alzheimer's disease for a moment to talk about another neurodegenerative disease, which is alpha-synuclein and alpha-synucleinopathy. So alpha-synuclein aggregates into Lewy bodies, and this is the main underlying neuropathology of Parkinson's disease, DLB, multiple system atrophy, a few other disorders. And it's also a very common co-pathology in Alzheimer's disease. And it certainly isn't uncommon in normal aging. And people that aggregate these pathologies, sure enough, they also have a great amount of heterogeneity in the symptoms that they present with. And, we wonder if they might have varying underlying disease trajectories. And so what's really interesting about the alpha-synuclein literature is there hasn't been a sort of defined consensus on how to stage the disease, like the way that there is with the Braak staging in Alzheimer's disease. So Braak also tried his hand in staging alpha-synuclein, and he basically came up with a system where you sort of have simultaneous buildup of alpha-synuclein in the olfactory bulb and the brainstem coming actually from the gut. So starting in the gut, making its way up to the brainstem, and you also have, and then throughout the rest of the brain. But then you also have, unrelated to that, pathology in the olfactory bulb. The Banner Institute has their own system, which I think is used more commonly. And in this case, alpha-synuclein starts in the olfactory bulb and the amygdala, and then spreads throughout the rest of the brain and goes down the brainstem and into the gut and peripheral nervous system. And more recently, mostly led, I would say, by Per Borkhammer at Aarhus in Denmark, there's been this proposal that, hey, maybe there's actually two different types, right, that might resolve this discrepancy in the staging systems. Maybe some people, they have alpha-synuclein that starts in the brain first in the olfactory bulb and makes its way down the peripheral nervous system. And some people have it starting in the gut, and peripheral nervous system makes its way up to the brain. We can have both of these things in the same disorder. And this is obviously a perfect context to apply a technique like sustain. And so that's exactly what we did. So this was a study that's led by a really phenomenal PhD student in the Biofinder group here at Sweden named Sophie Massenbrook. And so Sophie and Oscar Hansen and I sort of got our hands on several autopsy cases from the Banner Institute in Arizona. So these were people who had post-mortem Lewy body alpha-synuclein staining in a bunch of different regions of interest, which you can see here, some of them in the olfactory bulb, some in the brainstem, some in the limbic areas, and some in the cerebral cortex. Now, this data was based on these histopathological slides that look something like this. A pathologist gets a slide from a brain region, they look at it, and they basically give it a score from 0, no pathology, up to 4, very severe pathology. And so the question is, can sustain handle this type of data? And the short answer is no. Sustain, as it's been designed, is not designed to work with this kind of ordinal data. So thank goodness for Alexandra Young, who then said, hey, whatever, we can fix that, went to the lab and came back out relatively quickly with a new version of sustain that's designed to work with ordinal data that you might see in clinical scores, visual ratings, et cetera. And so that was published a couple of years ago and is now part of the online sustain ecosystem and can be used. So back to Sophie's project, she applied this new version of ordinal sustain to her alpha-synuclein data. Once again, she came up with these 3 subtypes. This is a very busy slide. We're going to just cut your focus, and I want to just kind of illustrate how we define these subtypes. And mostly it's what's happening at the beginning of the trajectory. So I want you to focus on 2 areas of the brain here, the olfactory bulb in sort of light blue and the brainstem in dark blue. So for this olfactory bulb limbic first subtype, we see early accumulation of Lewy bodies in the olfactory bulb that get really severe in these limbic areas like the amygdala, and we don't see any pathology in early stages in the brainstem. On the other end of the spectrum, we have people that have early pathology in the pons and the brainstem and none in the olfactory bulb. And even as the disease progresses throughout the brainstem, it remains, and even into the cortex, it actually remains fairly spared. There's not that much accumulation in the olfactory bulb in these people. And finally, there's something that's kind of in between. So there's people that have both olfactory bulb and brainstem accumulation. And so this sort of looks a lot like the different staging systems that have been introduced by different people. And you can see that there's all of these subtypes are fairly prominent. So people just kind of looked at their data and described what they saw, and they were all right. None of them are wrong. It's just there's multiple different trajectories, or at least that's what sustain seems to resolve onto. And so right, I mentioned that this sort of looks like the body first and brain first subtypes of Alzheimer's disease. And what's important is that there's theories that alpha-synuclein moves from the gut to the brain through the brain through the brainstem, and it does that through the vagus nerve, which connects the two through the peripheral nervous system. And so we had a select cases that had actually pathology measured in the vagus nerve. And so we're showing that here we actually want to focus only on these early stage cases, because after stage 12 or something, they start to blend a little bit. So we want to focus just on the early stage cases. And when we did, we found that sure enough, the people that had this early brainstem pathology, they had already a great amount of pathology in the vagus nerve. People that had this early kind of olfactory bulb and amygdala pathology in almost all cases had no pathology in the vagus nerve. And this middle group was sort of intermediate between them. So this really kind of supports this idea of having two different trajectories of alpha-synuclein. And we actually even took this a little bit further, looking at people that have at least 5 measures of peripheral Lewy body pathology, either in parts of the peripheral nervous system or the gut. And sure enough, we sort of always see the same kind of pattern where people with this OBT limbic first subtype, they basically have no pathology in the peripheral nervous system until pretty late in the disease, where they already have a ton of pathology in the brain. And these other groups, particularly the OBT sparing group, they tend to have already neuropathology in the peripheral nervous system when we see the earliest instances of brain pathology. So this is super interesting, right? I mean, we have a long way to go into understanding, what these subtypes are about. And there's a lot of evidence to suggest that they do have different symptoms. There's some data that we have that I'm not presenting here, this to show that they have different rates of disease progression. But I think it's just a really nice example of that. There's a lot of variation in these common umbrella diseases that we think about, like something as common as Parkinson's disease. So I want to end the talk I've been talking about subtyping this whole time. I just want to take a few minutes to talk about staging, which is also important. There's a few examples here. So we know we have a really good sense of how to stage alpha-synuclein, how to stage tau and Alzheimer's disease, but we have a lot of these rarer dementias like frontotemporal dementia or ALS that are much harder to characterize because there just are a lot less cases. So it's harder to get a gestalt feeling about the progression. And of course, there's also this relatively new phenomenon called late, which is actually quite common, characterized by buildup of TDP43 in limbic areas, but it's only been described kind of recently. So all 3 of these disorders involve the aggregation and spread of TDP43, but they have vastly different clinical syndromes and vastly different rates of progression and age of onset and all of that. And so there have been a couple attempts to kind of stage these different neuropathologies, but there's no consensus, especially here. I mean, this is a 2019 paper introducing late where they talk about there's actually various types of staging systems that have been introduced. So in collaboration with Eddie Lee and others at the University of Pennsylvania, we got our hands on several cases of late FTD and ALS that, again, had neuropathological data. And we basically applied sustain separately to each one. And this time we weren't trying to find subtypes. We just wanted to come up with a disease stage for each, a disease staging system for each one of these disorders. And that's what we did. This is obviously a very busy slide. I'm not going to go through these in detail, but you can see for each one of these staging systems, each one of these disorders, sustain came up with a staging system that described the really propagation of pathology from one part of the brain to another that really best fit the data. And this was really easy to do, really successful, I think, with ALS and late. With FTD, we really didn't have enough cases that were early or late stage to characterize this well, but we sort of did our best. And it was a very interesting exercise to then compare this sort of automated data-driven staging system with the ones that the neuropathologists had come up with just by looking at the data themselves. So here's an example of comparing our sustained stage of ALS with the ALS stage introduced by Brett Schneider and colleagues in 2014. You can see there's decent agreement, but their stage 1, 2, and 3, and 4 seems to have increasing sustained stage, but it's definitely not perfect. You can also see that the sustained stage is much more granular, right? So it's not quite as simple as this 1, 2, 3, 4. And I think most importantly, there's many cases that would be unstageable based on this ALS staging system that sustain nevertheless is able to sort of give us a stage 2. And we kind of found really similar results when we looked at the FTD staging and also the various different ways of staging late. Across all of these examples, we get pretty good agreement between automated and pathologist-based staging systems, but still many unstageable cases using the pathological staging systems, which are resolved by the sustained staging and obviously much more granular staging. And we started to realize something that we had a kind of nice staging system now for each of these disorders, and we can actually use that to our advantage for diagnosis, right? So what you're seeing here is we can apply each one of these models to a single person based on the score of pathology in various brain regions. And from that, we can get a probability that that individual falls onto this sort of staging system. So just to look in this panel A, you look at the probability that someone is ALS based on the ALS sustained model. And most of the ALS cases have a very high probability of being classified as ALS. And the other diagnoses, most of them had a very low likelihood. And we thought, well, this kind of looks like a classification algorithm. So we kind of combined this information in a machine learning logistic regression model, and we ended up with, whoops, looks like there we go. We ended up with a pretty good classification of balanced accuracy, out of sample accuracy, about 90%. And you can see some many train test splits. And this is quite interesting because we're sort of taking the pathologist out of the equation. We're taking the same data the pathologist is looking at, and we're seeing how well the model can recover the diagnosis that the pathologist then gave when they looked at the same data. And so it's actually kind of interesting to look at the cases where the model misses, because in many cases there's some discrepancy going on in the data. And it was really fun to sort of do a bunch of case studies to understand what's going on with those cases and why the neuropathologists did what they did. But we can also imagine this as something that could be used to aid neuropathologists when they do their diagnosis down the line. We can also stage neurodegenerative disease biomarkers. I'm looking at the time, and we're sort of running close to the hour, so I'm going to skip this one and just end with this beautiful work by Gemma Salvador, who's also part of the biomarker or biofinder group here in Sweden. And she has this beautiful study that's just been accepted recently in Nature Aging that's involving staging CSF biomarkers, right? So we can take a single CSF sample and we can measure various different biomarkers that are associated with Alzheimer's disease, right? So one is this A-beta 4240, which measures amyloid. And then we have tau, which has been phosphorylated in various phosphosites on the actual tau protein. And finally, this non-phosphorylated tau. And Gemma ran a sort of event-based model on this data and found that with extremely high confidence, there was this exact sequence that you're seeing here, that you very, very rarely saw 217 being abnormal before you saw 4240. You very rarely saw 205 abnormal before 217. This is interesting on a number of levels, because it tells us that there's a pattern and a sequence by which the tau becomes phosphorylated as the disease progresses. But it also has a lot of other very nice uses that I'm going to get into in a second. And so this is just the underlying data behind that sequence. Not going to go into this in detail, but it's important that what this is characterizing is how the biomarkers become abnormal and not, and not how they continue. So for example, here you can see that A-beta 4240 becomes abnormal very early, but then it kind of plateaus, and you're not getting any more information from this. In contrast, p-tau 217, it becomes abnormal fairly early, and it continues to become more and more abnormal over these different stages, and so forth. And what's very interesting is we actually tried to subtype this data, and we just didn't find subtypes. And that's for the sort of, haters out there that think you can find subtypes no matter what. No, we didn't find any subtypes in this data. This is just this is a sequence that fit all the data very, very well. And from this simple sequence, we were able to do so much, because we can then, in theory, take a CSF sample of someone, measure these 5 markers, and then assign them to one of these 5 stages. And what can we do with those stages? Well, we can first look at biomarker progression. This is really important for, once again, clinical trials, right? We find that in stages 1 and 2, longitudinally, this is all a longitudinal change, we're finding there's a longitudinal accumulation of amyloid beta, but that longitudinal accumulation begins to actually plateau and even decrease after stage 2. That means if you're trying to actually treat someone with an amyloid beta clearing drug, you should probably do it before they reach stage 3. Same thing with tau PET here. We can see that already in stage 2, it starts to become abnormal, and really it reaches the apex of its acceleration around stage 3. This is probably the optimal time to treat someone with these upcoming tau-related drugs. And so you can imagine it's not that expensive to get a CSF lumbar puncture. If you want to just screen people based on doing that, it would be way more inexpensive than using PET. We can also do really good differential diagnosis with this information, right? We can, unsurprisingly, do a really good job classifying whether someone is amyloid positive or tau positive in unseen data. And by extension, we can also do a pretty good job differentiating Alzheimer's disease from another neurodegenerative disease, something that could be very helpful in a memory clinic. And finally, we can do some pretty nice cognitive forecasting, right? So someone who's in stage 4 or 5, if they have MCI, on average, or I should say 50% of the people that were stage 4 or 5, based on the CSF staging, converted from MCI to dementia in about 2.3 years. And if you look at people who are cognitively unimpaired, well, even those that didn't have a cognitive diagnosis, if they were stage 4 or 5, about 50% of them declined to MCI in about 4 years. So there's so much information we can get from a single CSF measurement. And there's so much potential in this and something we're exploring. And we're now actually trying to translate this to plasma so we can get a blood test and hopefully get most of this information as well. I'm going to skip this T-sustain thing for now and just sort of get to the summary. Many diseases likely encompass subtypes. I hope I've convinced you at least of that. And I do think that subtypes can have a bearing on clinical outcomes that are relevant for trials as well as in the memory clinic. Now, I didn't talk much about what subtypes can teach us about disease biology. There is a lot to talk about in that context, and I'd love to talk to you about that if you're interested offline. And the position that I'm sort of opening now is going to be based more on that. And I didn't really talk too much about the caveats either. For example, how much data you actually need for this to be successful. But if you're really interested in using Sustain, I would love to talk to you about this. And you don't even need to talk to me if you don't want to, if you're shy or you don't like me or whatever, because there's also our Git, or I should say the Pond GitHub for PySustain, which has all the code you need. It has a bunch of tutorials and sample notebooks you can just copy and paste. It's got a bunch of links, including to this YouTube channel that has an entire 2-hour Sustain tutorial, as well as a bunch of smaller vignettes about certain aspects of the Sustain model. And I invite you to check out that to learn more. And with that, I will thank you for your attention and thank all the people in the Swedish Biofinder study that provided most of the data for a lot of what you saw here. And I'm specifically highlighting some of the people that either led or co-led the studies or provided the data that we needed to actually do these studies. But of course, there's so many other people that contribute in other ways, like data collection, funding, and of course, collaboration. So thanks so much for your time. And if you have any further questions or you want to just talk shop about Sustain, I'm very open to chatting with any of you. Please email me, set up a chat, or we can just continue the conversation offline.
Thanks a lot, Jake. Thanks for joining us. I really appreciate you making the effort. And what a great talk. That was really phenomenal, very clear, and really exciting stuff. Thanks a lot. Take care.
Thanks so much.